Hello everyone, and thank you for joining this AJMC TV peer exchange video program based on evidence-based decisions to improve quality and regulate costs. This program is part of the Oncology Stakeholder Summit series and features key insights from a distinguished panel of experts, each well known in our professional community. I'm Dr. Peter Salgo. I'm a professor of medicine and anesthesiology at Columbia University and an associate director of surgical intensive care at the New York Presbyterian Hospital in New York. I'm joined today by Dr. John L. Fox, Senior Medical Director and Associate Vice President of Medical Affairs at Priority Health, Grand Rapids, Michigan. Dr. Ira M. Klein, National Medical Director, Clinical Thought Leadership, Office of the Chief Medical Officer, Aetna Incorporated, Hartford, Connecticut. Dr. Michael Kaloje, National Medical Director for Oncology Strategies, Aetna Incorporated. Dr. Brian Loy, Physician Lead, Cancer, Humana, Louisville, Kentucky. Dr. Erwin W. Tischler, National Medical Director, Oncology, Cigna in Phoenix, Arizona. And joining us remotely will be commentary from Dr. Christopher Sweeney from the Lank Center for Genitourinary Oncology at Dana-Farber Cancer Institute. Gentlemen, and it is all gentlemen here, thank you again, and why don't we just get started. We're going to be discussing data and evidence from studies. Now, they're evaluated by multiple stakeholders here. There are regulatory agencies, there's physicians, payers, patients, and the drive for generating quality data includes safety, efficacy, and of course, rising healthcare costs. Although I'd like to say for the record, they're rising less steeply than many feared they would be rising at this moment. So that's very interesting. Now, unless a treatment regimen is deemed valuable by all the stakeholders, chances of its clinical success, and I mean by that clinical adoption, are going to be low. So what would distinguish data collected from interventional versus non-interventional oncology studies?
Sure, I'll take that one. RCTs that are done as a prerequisite to FDA approval are kind of getting the foot in the door. That RCT and FDA approval will cause us as a health plan to cover that drug. But what we as payers, what patients, what providers would like to know really is what optimal therapy is. And optimal therapy is oftentimes not distinguished in clinical trials, and that's where comparative data would help us define, given all the options that are available, what is best for patients, what's best for payers, what's best for providers.
So would it be useful to you to have this non-interventional data, or is all data useful to you?
Well, I think that the data that we're initially presented with is inadequate to help patients and providers and payers decide what optimal therapy is.
So let me ask the $64, trillion question by the time we're all through with this. I'll send it your way. How can data collected from non-interventional studies, or can data collected from non-interventional studies ever replace trial data? And if the answer is yes, why?
I don't think it can ever totally replace clinical trial data. I think that still remains the gold standard. However, randomized clinical trials, what we're referring to, is really not real-world situations because they are so focused on a particular data set, and it doesn't necessarily translate well. The non-interventional, the data review, the metadata analysis, I think can go a long way in the areas that we're looking at in personalized medicine and further expanding these particular trials.
I don't want this to just sit there unchallenged, guys.
So let me comment. I think that there's another factor that is going to dramatically change how we view data collection and data quality, and that is, in fact, the personalized medicine space. So the historical approach to drug development and FDA regulatory approval and then adoption and practice is going to change because there are so many genotypic possibilities, so many phenotypic possibilities, so many actionable mutations in a patient, that personalized medicine is going to force us to reconsider what good data looks like and what really actionable data looks like. You just can't do a trial for every single question that's going to come up.
But I know you have something to say, so why don't you get that in first, because I'm still troubled by something he said. So unless you're troubled by the same thing, I'll ask it in a moment.
Well, I think Mike is right, and yet we can't do a trial on everybody, and yet how do you know when information is enough. For example, BRAF V600E mutations in melanoma, there's a great drug for that. But that same BRAF mutation in colon cancer connotes failure to respond to that drug. So when do you have enough information?
I mean, right, you're sort of getting at what troubled me. If we will all agree that the double-blind controlled studies don't answer everything, what do we have that's any better? Isn't it the gold standard? Isn't that all we've really got? Or am I missing something here?
So in fact, if you look at the history of oncology, the standard paradigm is FDA approval, randomized clinical trial, very controlled population, and then we figure out how it really works. And that's what has happened historically in oncology. Now, what has gotten in the way of that now is the ridiculous cost of care, right? So the drugs are so expensive that even when you're in a terribly desperate situation, and there's a lot of terribly desperate situations, it's really hard to just sort of wing it. And so I think providers are challenged in that circumstance today. Personalized medicine, molecular medicine, somatic mutation analysis further mutates that paradigm. So now we've got a situation where there's a reason to try something. So now we're going to have to redefine the parameters of, is that reason good enough?
And we've also got the patient-reported outcomes, right? Which is another variable that when I was in medical school, we never asked.
Yeah, I mean, I think I can comment on that, and I know Brian has a comment as well. You know, that's just going to be another wrinkle in the whole mix that includes the very narrow segmentation of patient populations because of the genetic fitting them into this somatic mutation, that somatic mutation. So I think we need that information, but I think it's only going to be part of the mix. And I think we can't misinterpret that information like we've done in other mandated types of data collection that's patient-centered, such as consumer assessment of health plan satisfaction, things like that. Those have been very tough to figure out. I think in specific oncology, you know, we're always going to be picking efficacy, then we're going to look at toxicity, and I think in that tolerability realm, we're going to have to get some patient information. But Brian, you wanted to comment.
My comment was more about, I don't know that we even have to answer the question of either or. I think we've got to be selective, and there's going to be a series of trade-offs when we elect to do a randomized clinical trial versus something other. But if you start to think about, so where are we today? And to Mike's point earlier, if we're not getting the value out of the dollars that are being spent for the care that's being delivered, you know, we are in a position to be able to accumulate some of that data and begin figuring out, so where is the variation and how can we begin studying that variation that exists?
Well, that brings us right to this.